Okay, guys, I hope everything is going well for you. We've had a pretty rainy week this week. We're going to finish up chapter 11 today, and then on Thursday and Friday, you are going to be completing your lab handouts for lab exercise 14 and 15. Also, tomorrow on Thursday, you are going to be having a quiz on a Google form over lab exercises 12 and 13. So make sure you do that first, and then I will be posting the lab pages for lab exercise 14 tomorrow, and then lab exercise 15 pages and handout will post on Friday, and you should make sure that you get those completed. Again, we will be having another quiz over lab exercises 14 and 15 next Thursday, and we will also be having a quiz over chapter 11 on next Monday. And then remember, after next week, it will be spring break, so we won't be doing any work. I won't be assigning any work on Google Classroom April the 6th through 10th. Again, I know I'm not having you to turn in anything for me related to the labs, because if we were at school, I would not be having you to turn anything into me for the labs. However, just because I'm not asking you to turn something in doesn't mean you don't need to do them. Please make sure that you're reading over the lab handout or over the lab pages. You are printing out and completing the lab handouts for these lab exercises. Again, it's very important that you do that because that is going to give you exposure to these muscles and these diagrams, because you will be having another lab exam. And if we are back at school, you won't have anything to use for that lab exam. And so you will have to study those lab handouts in order to take that lab exam. I may just make it pencil paper, since pretty much all the lab stuff for this lab exam at this point has been just pencil paper stuff. I may just make it pencil paper and not ask you to look at any models and identify anything on models, because you haven't had the chance to look at them. But, if we are back at school, when you have this lab exam, it will be pencil paper, and you will have to have completed and studied all of those lab handouts in order to be able to label the diagrams that I will ask you to label on the lab exam. So please be completing those lab handouts. If you can't print them out in color at home, then again, those copies of those lab handouts can be printed out for you in color at school. Even though I'm not there, I have emailed them to Miss Emberton and told her that if you came by to pick them up, you would need them printed out in color. You just might want to remind her of that, but if you do need them printed out for you in color, then you can go by the high school, and Miss Emberton has those in her email, and just remind her that you need them printed out in color. All right, so we're going to finish up with chapter 11 today. Hopefully it won't take very long. We have been looking at how an action potential is generated, and now we're going to look a little bit more at how the action potential is transmitted from one cell to another. Again, we've already looked at this when we talked about the muscle being stimulated by the nerve cell in chapter 9, so a lot of this is just going to be review. Some of it will be new, but some of it will be review. So again, remember that when we were talking about the connection between the muscle and the nerve cell, we call that the neuromuscular junction. When we talk about the connection between a nerve cell and another nerve cell, we call that a synapse. So it is the junction mainly between two nerve cells. It could also be the junction between the nerve cell and the muscle cell, but we're going to call it a synapse here between two nerve cells. There are two types of synapses. Before we talk about the two types of synapses, there are two types of cells in a synapse. The presynaptic and the postsynaptic. We've already used this before as well. The presynaptic is the cell, when we discussed it, the presynaptic cell was the nerve cell, okay? But again, it is the cell that is sending the signal. It is sending out the signal. Many times that's a neurotransmitter. And then the postsynaptic cell is the cell receiving that signal. So in our neuromuscular junction, the presynaptic cell is the neuron and the postsynaptic cell is the muscle. But if we're talking about a synapse, a junction between two nerve cells, then the nerve cell sending the neurotransmitter, sending the signal, is the presynaptic, and the nerve cell receiving the signal is receiving the neurotransmitter is the postsynaptic. Again, there are two types of synapses. The ones that we talked about the most are chemical synapses, but there are also electrical synapses. So we're going to talk about electrical synapses first. Electrical synapses are going to be junctions between two cells that are connected by gap junctions. In this gap junction, you have these tunnel-like protein tubes called connexins. And those connexins in this gap junction allow sodium ions to freely move from one cell to another, and so therefore the current or the action potential freely moves from one cell to another. You're going to find these in both cardiac muscle and smooth muscle. Remember those intercalated discs in cardiac muscle are gap junctions. Remember that our smooth muscle, especially that visceral smooth muscle, also some of that visceral smooth muscle has those gap junctions. And so you have these electrical synapses where these sodium ions can move freely from one cell to another, which allows the action potential to move freely from one cell to another. Again, pretty much we find these in cardiac and smooth muscle, that visceral smooth muscle, because those muscles are autorhythmic. Okay, so it's important for groups of cells to be able to contract together in unison. In your heart and in places like your digestive tract. So therefore they're autorhythmic and they're going to have these electrical synapses with those gap junctions. And this is just showing you here, this is cardiac muscle. You can tell by the branching cells, the intercalated discs, and the striations. And you can see here, this is a gap junction. This is one cell, this is another cell, and this is the tube-like tunnel of protein called a connexin. And so sodium ions are able to freely move through that connexin, and when sodium ions move, you know that's how we get an action potential. So action potential's current is also able to freely move from one cell to the other to allow these cells to contract together in unison to cause your heartbeat in this example. Okay, the chemical synapses are the ones that we talked about in Chapter 9 with the nerve and the muscle cell, that neuromuscular junction. And we also have lots of chemical synapses between nerve cells. Chemical synapses between nerve cells. Again, you have a presynaptic terminal, which is going to be of the presynaptic cell, the cell that's sending the signal, and then you have the postsynaptic membrane or the postsynaptic cell, which is the cell that's receiving the signal. Again, the presynaptic is going to be a neuron. The postsynaptic can be a muscle cell, it can be another neuron, or it can be a gland cell. Remember that these neurons are going to be stimulating our skeletal muscle. That's going to be that somatic nervous system, contains those motor neurons that are innervating our skeletal muscle. Also, we have those autonomic neurons that are stimulating our smooth muscle, as well as our glands. Also, these chemical synapses can be, a nerve cell sending a signal from one nerve cell to the other. So the presynaptic is always going to be a neuron. The postsynaptic can be a neuron, a muscle cell, or a gland cell. And then your synaptic cleft, again, remember, is going to be the space between the two cells. And remember that action potentials cause neurotransmitters to be released from this synaptic terminal, again, or from this presynaptic terminal. Again, remember that neurotransmitter is the signal, it's the message. So the neurotransmitter is released released when an action potential goes through that presynaptic terminal. Remember that the action potential is going to cause calcium ion channels to open. This will allow calcium ions to enter that presynaptic terminal, and then those calcium ions fuse with synaptic vesicles and signal those vesicles to release their neurotransmitters into the synaptic cleft. Remember then that neurotransmitter diffuses across the synaptic cleft and binds to receptor proteins on the ligand-gated ion channels. Generally, especially if we're talking about a muscle cell, the neurotransmitter acetylcholine binds to the receptor proteins on the ligand-gated sodium ion channel and that opens that sodium ion channel, that ligand-gated sodium ion channel, and allows sodium ions to move through. So we can see this here in this picture, and this is describing the same thing. An action potential comes down that presynaptic terminal of that presynaptic neuron, and it stimulates, these are voltage-gated calcium ion channels, so that action potential causes those voltage-gated channels to open and allow calcium to come in. Calcium comes in and signals these synaptic vesicles with those little blue triangles, which are acetylcholine. Those vesicles fuse with the membrane, and they cause that acetylcholine to be released into this synaptic cleft, which is the gap. Then those little blue triangles, that acetylcholine, bind to these ligand-gated sodium ion channels in this case. When they bind, it opens that sodium ion channel and allows sodium to move into this postsynaptic cell, of this postsynaptic membrane. And again, then that sodium is able to stimulate an action potential in this postsynaptic cell. Again, once we have stimulated that action potential, we're not going to necessarily keep causing more and more action potentials. So you're going to have something to get rid of these neurotransmitters. And for different neurotransmitters, there are different enzymes that are going to get rid of them. Two big neurotransmitters that we'll be talking a lot about are acetylcholine and norepinephrine, also called noradrenaline. Epinephrine and adrenaline are the same thing, and then norepinephrine, noradrenaline are also the same things. And we'll be talking more about adrenaline/epinephrine, and we'll be talking about norepinephrine and noradrenaline in future lessons. So anyway, we've already learned this in anatomy last year. Acetylcholinesterase, or ACHase, is what's going to break down that acetylcholine, that ACH. It will split that acetylcholine into acetic acid and choline. The choline will be recycled within the presynaptic neuron. It will be reabsorbed and then recycled by the presynaptic neuron. The acetic acid will move on into the bloodstream to be carried away as waste. For norepinephrine, there is also an enzyme that breaks down norepinephrine. It is called monoamine oxidase. So MAO, or monoamine oxidase, is the enzyme that will break down norepinephrine, just like ACHase breaks down acetylcholine. Another thing that can happen to the norepinephrine, if it's not immediately broken down by the monoamine oxidase, they can actually be absorbed into the bloodstream, and it will be carried to the liver and kidneys, and there it will be broken down by again, the MAO, the monoamine oxidase, or another enzyme called catechol-O-methyltransferase, or COM, C-O-M. So there are actually two different enzymes that can break down the monoamine oxidase in the synaptic cleft. After it diffuses away from the receptor on that ligand-gated ion channel, in that synaptic cleft, it can be broken down by the MAO. If it is absorbed into the bloodstream, then it will go to the liver and kidneys and be broken down either by MAO or COM. So again, there's multiple pathways that norepinephrine by which it can be broken down. Again, this is a picture showing us what happens to the ACH. Again, after the ACH has bound to the ligand-gated channel and it has opened it and allowed the sodium in, then it will diffuse away from that channel and it will be broken down by the ACHase here in the synaptic cleft. The choline will be reabsorbed into the presynaptic membrane, the presynaptic cell, and be recycled to make more ACH. The acetic acid will diffuse away as waste. Here we see what happens with the norepinephrine, which we're going to call NE. NE is norepinephrine. Okay, once it has bound to the receptor of the ligand-gated ion channel and caused it to open, then it will unbind it and diffuse away from that receptor protein, and here in the synaptic cleft it can be broken down, or actually it looks like it's probably being broken down by MAO in the presynaptic cell. So basically what's going to happen is the norepinephrine will be reabsorbed into the presynaptic cell, broken down by MAO, and then or else it can go on into the bloodstream and go to the kidneys and liver and be broken down by MAO or COM in the kidneys and or liver. Okay, something important for us to know that about the receptor molecules on those ligand-gated ion channels in these synapses, that certain neurotransmitters will fit certain receptors on these ligand-gated ion channels. A neurotransmitter will only bind to receptors that it fits. Okay, so therefore neurotransmitters can only affect cells that have those receptors on those ligand-gated ion channels that are specific for that neurotransmitter. So you can have, let's say you can have norepinephrine being secreted into the synaptic cleft. If that postsynaptic membrane does not have receptors on its ion channel specific for norepinephrine, you can secrete norepinephrine into that synaptic cleft all day long and it's not going to cause anything to happen if that membrane does not have receptors for that norepinephrine. Okay, so the neurotransmitters are specific. They fit only one receptor, and if that postsynaptic membrane does not have receptors for that neurotransmitter, then there will be no effect on that postsynaptic cell. The neurotransmitter will affect only cells that have receptors for them. Also, another thing to know about these neurotransmitters, some are excitatory, some are inhibitory. So for some cells, let's say for our skeletal muscle cells, okay, acetylcholine causes contraction, so it would be considered excitatory. Okay, norepinephrine would be inhibitory. So it all depends upon which neurotransmitter is being released into that synaptic cleft. If acetylcholine is being released, that causes depolarization of the muscle cell, and that causes an action potential, and it causes muscle contraction. But if norepinephrine is being released into that synaptic cleft, then it doesn't cause depolarization. It actually causes hyperpolarization. It's going to open up those potassium ion channels and let potassium come out of the cell, which is going to cause hyperpolarization of the cell, and it's going to be inhibitory, and it will not cause muscle contraction. So again, it all depends upon which neurotransmitter you have being released into that synaptic cleft as to whether or not there's going to be an effect, and if there is an effect, what type of effect it's going to be, excitatory or inhibitory. Also, some neurotransmitters, such as norepinephrine, can actually attach to the presynaptic cell. So the cell that's releasing them, it can actually go back and attach to that cell and cause that cell to stop releasing it. So it actually modulates its own release. So if that presynaptic cell, that nerve cell, is releasing norepinephrine can actually go back and, instead of diffusing across the synaptic cleft and binding to the postsynaptic cell, it can actually go back and bind to that presynaptic cell and cause that presynaptic cell to not release any more norepinephrine, so controlling its own release. Neurotransmitters, again, are chemical messengers secreted by neurons. They are stored in synaptic vesicles. Action potentials stimulate their release into the synaptic cleft by exocytosis. They bind to specific receptor proteins on ligand-gated ion channels on postsynaptic membranes, and they evoke a response in the postsynaptic cell. They cause an effect on the postsynaptic cell. So all of that is the definition of a neurotransmitter. We can have neurons that secrete multiple kinds of neurotransmitters. So you could have the same neuron releasing both acetylcholine and norepinephrine, or acetylcholine and adrenaline. You can have it releasing both of those, just maybe at different amounts or different times. There are at least 100 different types of neurotransmitters. Acetylcholine neurotransmitters are the best understood. But we also have biogenic amines. There are two kinds of those, catecholamines and indolamines. Norepinephrine, epinephrine, and dopamine. I know you've heard of that before. That is actually a neurotransmitter that causes you to, when you take a risky behavior, it is released and causes you to feel good. So that is associated with risk-taking behaviors, because people who like to take risks, when they take a risk, their brains release dopamine. Their nerve cells release dopamine, which causes them to have a good feeling. So that's why they like to take risks. These are the dopamine, norepinephrine, and epinephrine are called catecholamines, and they come from the amino acid tyrosine. So the amino acid tyrosine that's in proteins is used to make dopamine, norepinephrine, and epinephrine. Indolamines come from the amino acid histidine. So again, histidine in proteins can be used to make histamine and serotonin. Another amino acid that's used to make these is tryptophan. So histidine and tryptophan and tyrosine are all amino acids that can be used to make these catecholamines and indolamines. Another type of neurotransmitter are amino acids themselves. Some amino acids, like glycine and glutamate, can actually be used by cells as neurotransmitters, okay? So again, your tyrosine, your histidine, and your tryptophan themselves, they are not neurotransmitters, but they can be used to make dopamine, norepinephrine, epinephrine, histamine, and serotonin, okay, which are neurotransmitters. Then there are some amino acids like glycine and glutamate that can themselves act as neurotransmitters. They can bind to receptors on the postsynaptic membrane and cause an effect on that postsynaptic cell. Another example of a neurotransmitter, and again, these are just a few of the examples, there are over 100 different types, are purines. Purines are actually the nitrogen bases that are used to make DNA and RNA nucleotides. They can also act as neurotransmitters. An example, ATP can act as a neurotransmitter. Neuropeptides. These are short chains of amino acids, about 10 to 40 amino acids long, that can also act as neurotransmitters, again called neuropeptides because peptides, okay, those are the short parts that make up a protein, which are made of amino acids, and then neuro because they are acting as a neurotransmitter. You've heard of endorphins. Endorphins are chemicals, for example, like when you exercise, endorphins are released by your neurons, and they cause you to have a good feeling. So they affect your brain in that they make you have a good feeling. Sorry about that. We're going to keep going. Anyway, so endorphins again are neurotransmitters. They are neuropeptides. Continuing on, the last example that we'll look at are gases and lipids. Some gases, such as nitric oxide gas, guys, this is the gas that you take, that you get at the dentist office, nitrous oxide. Okay, that gas that you breathe at the dentist office, it is a neurotransmitter, and that's why it has that effect of kind of calming you down and making you kind of feel sleepy and a little bit out of it, because it acts as a neurotransmitter. Then we also have lipids such as endocannabinoids. These are neurotransmitters. They actually bind to the same receptor as the THC in marijuana. So this is why marijuana gives you that feeling, or not. I don't want to say you because I hope you don't use that. But this is how marijuana gives people that feeling when they smoke it, because the THC in the marijuana acts as a neurotransmitter. It binds to the same receptor that these endocannabinoids bind to, and has effects on that postsynaptic cell. Again, neurotransmitters can be excitatory or inhibitory. If they cause depolarization, which means they're opening sodium ion channels and allowing sodium into the cell, they are excitatory. If they cause hyperpolarization, if they bind to receptors and cause potassium, ligand-gated potassium ion channels to open to allow potassium out of the cell, then they are going to be inhibitory, because when potassium comes out of the cell, that causes hyperpolarization, and more than likely an action potential won't be produced. So again, we can have a combination of neurotransmitters being released into the synaptic cleft, and it's the combination of neurotransmitters and the receptors that they bind to that are present on that postsynaptic cell that is going to cause either depolarization or hyperpolarization of the cell. If depolarization is caused, this results in an EPSP. If hyperpolarization is caused, this results in an IPSP. And we'll talk about an EPSP and an IPSP here in just a second. But again, we have to realize that, you guys, there's not just one neurotransmitter being released into that synaptic cleft. You're going to have multiple neurotransmitters being released into the synaptic cleft by the same neuron or by different neurons that are synapsing with this cell, with this other neuron, or this postsynaptic neuron, or this postsynaptic muscle cell. You're going to have I don't want to say with a muscle, because mainly with a muscle it's going to be acetylcholine, period. But let's say we have a synapse where we have a presynaptic neuron and a postsynaptic neuron. You're going to remember, these neurons can release more than one neurotransmitter. So you may have multiple neurotransmitters being released by that presynaptic neuron. Well, what's going to happen to that postsynaptic neuron depends upon which neurotransmitters are being released by that presynaptic neuron, and also which receptors are on that postsynaptic neuron membrane. Okay? So it's the combination of the neurotransmitters and the receptors that are going to determine whether we get depolarization and an action potential, or hyperpolarization and no action potential. Okay, again, we said depolarization results in an EPSP, hyperpolarization results in an IPSP. Okay, what is an EPSP? An EPSP is an excitatory postsynaptic potential. So again, EPSP, excitatory postsynaptic potential. What this is, you guys, simply put, is when depolarization occurs and the postsynaptic cell is stimulated, it's excited, depolarization happens and an action potential happens, and we get stimulation of that postsynaptic cell. That means that potential that we produced was an EPSP. Again, if there is enough depolarization, we do reach threshold, we get an action potential, and the cell is actually stimulated. Again, this EPSP may not end in an action potential if you don't get enough depolarization, but if you get enough depolarization, if you get enough sodium ions in to get up to negative 55, then you're going to get an action potential and you're going to get stimulation of that cell, for example, a muscle cell to contract. Okay, so an EPSP is what causes that to happen. Neurons that release neurotransmitters that cause EPSPs are called excitatory neurons. But then we also have IPSPs, which are inhibitory postsynaptic potentials. Again, these IPSPs result in hyperpolarization, and so we get an inhibitory response. And the neurons that are releasing these are called inhibitory neurons. So neurons that release neurotransmitters that cause IPSPs are called inhibitory neurons. We also have neuromodulators. We're not going to talk about these very much, but these are substances released from neurons that can influence the likelihood of an action potential being produced in a postsynaptic cell. You guys, this is how things like cocaine, amphetamines, and antidepressants work. They are neuromodulators. They are not neurotransmitters. Like the THC in marijuana binds to the same receptor as the endocannabinoid, so it is probably acting more like a neurotransmitter. Okay, these neuromodulators are not neurotransmitters. They are not binding to the receptors. They are causing an increased release of the neurotransmitter norepinephrine, and they are blocking the reuptake of these neurotransmitters like norepinephrine and serotonin. So again, they're not acting as neurotransmitters. They're not binding to the receptor proteins on those ligand-gated ion channels on that postsynaptic cell. They're not binding to the receptor. What they're doing is that they are blocking, they are having an effect on the presynaptic cell, on that presynaptic neuron. They are blocking it from reuptaking that neurotransmitter once it has been released. They are blocking it from taking it back up. And they can actually cause it to increase the release, to release more of the neurotransmitter. So rather than binding to the receptor on the postsynaptic cell, they're not having an effect on the postsynaptic cell. They are affecting the presynaptic cell. And by affecting the presynaptic cell, that causes a more of an effect in the postsynaptic cell, because they're increasing the release of the neurotransmitter from the presynaptic cell, and they are also blocking the presynaptic cell from taking that neurotransmitter back up once the action potential has been caused. They are blocking it so that you keep getting action potentials in that postsynaptic cell, which is what causes the feelings that you have as a result of taking some of these neuromodulators. Okay, we also want to talk about spatial and temporal summation. Basically, a single potential, again, a single postsynaptic potential is not enough to cause an action potential. Again, we kind of talked about this a little bit yesterday. One little graded potential is not necessarily enough to reach threshold to cause the action potential. So when we talk about a graded potential, we talk about a postsynaptic potential. We're kind of talking about the same thing here. A graded potential, a single graded potential, a single postsynaptic potential may not be enough to reach threshold and cause the actual action potential that stimulates the cell. Many times it's going to take many of these graded potentials, many postsynaptic potentials to combine together at that trigger zone, to summate at that trigger zone to reach threshold to cause an action potential. So there's a couple of different ways that summation can happen with these graded potentials, with these postsynaptic potentials. We have spatial summation and temporal summation. With spatial summation, we have two graded potentials or two action potentials, I should say, that are arriving simultaneously. So we have an action potential, one arriving here from this neuron. We have from this yellow neuron here, and then we have another action potential arriving from this neuron here in this yellow one. So we have two action potentials. Neither one of them are enough to reach threshold to give us an actual action potential, neither one separately. But if they both arrive together simultaneously at the same time in this postsynaptic neuron here, then they add together here at this trigger zone, where this axon hillock and this initial segment come together, this trigger zone. Both of those smaller graded postsynaptic potentials, both of those graded potentials together separately are not big enough. They don't cause an action potential. But when they add together here at this trigger zone, they do add up and reach threshold here and gave us an action potential. Okay, so that's what we're talking about with spatial. They're arriving together in the same space, the same neuron at the same time. So spatially, they arrive together at the same neuron at the same time, and they give us an action potential. Temporal summation, we are referring to two, again, postsynaptic potentials, two graded potentials that are arriving together, one right after the other. Okay? So it's not the fact that these are arriving at the same time right here at the same cell and adding together. Okay? So spatially, they're very close together to one another. Here, they're both arriving at the same place. Here they're arriving at two different places in the same cell. Okay? Here they're arriving, the same neuron is giving these graded potentials, these postsynaptic potentials, but it's just doing one right after the other. So neither one separately is enough to cause an action potential, but the two arriving one right after the other in very close succession we reach threshold and we get that action potential. So that's the difference between spatial and temporal summation. Another thing we have to understand, guys, is that look at this. If we go back and look here, look at spatial summation. You have more than one. You have two presynaptic neurons stimulating this postsynaptic neuron. Here, we see that each neuron may be sending more than one postsynaptic potential. So what we actually end up with is we have one postsynaptic neuron that is receiving many postsynaptic potentials from many different neurons. It's receiving postsynaptic potentials from many different neurons. It's receiving many from this neuron here. It's receiving more than one. So we end up with many EPSPs and IPSPs. Some of these neurons are sending EPSPs. Excitatory postsynaptic potentials. We have others that are sending inhibitory postsynaptic potentials. So many neurons stimulating this one postsynaptic neuron. And so what we have to understand is that there's many excitatory and many inhibitory. And it is the sum. It is the combination of all these EPSPs and IPSPs that determine what this postsynaptic neuron is going to do. If all of these IPSPs and EPSPs summate together to cause overall threshold, then we're going to get an action potential and we're going to get stimulation. If the IPSPs are going to cancel out enough of the EPSPs that we end up not getting threshold, then this cell won't do anything. So again, we have to understand that lots of presynaptic cells are trying to stimulate or inhibit this postsynaptic cell. And it's the sum of all these together. If the EPSPs are enough to cancel out the IPSPs, so we basically have more EPSP than IPSP, then we're going to reach threshold, we're going to get an action potential, we're going to get stimulation, we're going to be excited. If the EPSPs are not enough to cancel out all the IPSPs, then the IPSPs will dominate and it will be inhibitory and you won't get an action potential and nothing will happen in this cell. You won't reach threshold. So again, we have to understand there's lots of cells that are trying to excite or inhibit this postsynaptic neuron. We have to understand that there's lots of different neurotransmitters that may be being released by these presynaptic cells. And so it's the combination of the neurotransmitters that are being released, the receptors that are on this postsynaptic cell, as well as it is the combination of, are we getting inhibitory, are we getting EPSPs and IPSPs? Okay, again, what determines that? It's the neurotransmitter. If it's an excitatory neurotransmitter, then we're going to get an EPSP. If it's an inhibitory neurotransmitter, then we're going to get an IPSP. Okay, last thing that we want to talk about today are different pathways. We're not going to talk about all 4 of these. I'm going to focus on these 2. The organization of neurons in your CNS can be different as well. Okay, if we have a convergent pathway, this is where we have many neurons that are synapsing with a smaller number. So a large number is converging into a smaller number. This is how your brain synthesizes data, okay, during your thoughts. This is how you synthesize data in your brain. The opposite of that is a divergent, where we have a small number of neurons that synapse with a larger number of postsynaptic neurons. And this is how important information is transmitted, is gotten out to lots of different parts of your brain. So basically, convergent, I think of synthesizing data. Okay, with divergent, I think of spreading out. I think of transmitting information to lots of parts of my brain. So here's our convergent. A large number converge with just a few. Okay, this is synthesizing something. With divergent, it's just the opposite. You have a small number that synapses with a large number to send information out to a lot of different places. And then we're not going to worry about the reverberating and the parallel after discharge. Okay, please let me know if you have any questions. Email me, put a comment on Google Classroom. Otherwise, I hope you have a great rest of your day. Let me know if you have any questions. This is it for Chapter 11. We will be having a quiz over Chapter 11 on Monday. And then don't forget a quiz over lab exercises 12 and 13 tomorrow before you do your handout for lab exercise 14. Again, have a great rest of your day. Stay healthy, and we'll talk to you later. Bye-bye.